So thank you so much for joining us today. I'm Dr. Jo Bichell. I am a neuroscientist and I'm also the mother of a 22-year-old son with Angelman syndrome. I am really having fun with this jointly produced program called Angelman Updates that's being produced by both FAST and the ASF. About the scientists who are doing the most exciting cutting edge research in the Angelman field. And so today I am interviewing Dr. David Segal, who is one of my favorite people in the field. He is just an amazing guy who has not only done incredible research and really cutting edge research, but he's also run marathons for Angelman causes. And if I recall, one of them was actually on the Great Wall of China. So, I mean, my God. Anyway, so thank you very much for doing the interview today. And so if you could just introduce yourself.
Well, thank you for inviting me to be on the show today. And I'm David Segal. I work at UC Davis in the Genome Center, the Department of Biochemistry, and also in the UC Davis Mind Institute. And I've been involved in Angelman syndrome research now for a number of years.
So that was pretty vague. We want to know exactly what have you been doing in Angelman research? Well, should we start with what have you been doing lately, maybe? Or should we start with what did you do? Let's start with what have you been doing lately with Angelman research?
Well, actually, I think to get to where we are today, it might be good to kind of give a little background. So when I first got involved in Angelman syndrome many years ago, I was contacted more or less out of the blue by a group of, this was before Facebook, so this was a Google group of scientists and concerned parents who had some kind of a group and contacted me, had read some of my papers, and wanted to know if the technology that we worked with could be useful in the treatment of Angelman syndrome. And back then, I had never even heard of Angelman syndrome. So I had to look it up on that vast repository of human knowledge, Wikipedia. And then, I kind of got the idea of what they were talking about. Um, but I said, I don't know if this approach will be useful because it's not clear that, since this UB3A gene had not been expressed, if we could do something about it, much later in life. I was very fortunate to be part of what came to be known as the FIRE team, which was a team of scientists that FAST put together, just dedicated to working on Angelman syndrome. And importantly, the team was supposed to be able to discuss openly the work that they were doing with each other, so that it was more of a collaborative effort rather than a competitive effort uh, in trying to do this research. So that was a very exciting thing to be part of. And so we had groups like Ann Anderson and Ed Weber that were working on kind of electrophysiological kinds of aspects of Angelman. And then with Scott Dindo and myself, we were more on the genetic side of things. And so we were all working on different aspects of developing therapeutics. As many of your listeners will know, Scott Dindo went on to really take on the ASO challenges. And the work that I was doing was trying to make transcriptional regulators that could turn on UB3A. And more or less, we're still working on that today.
So how did that fire team work? Did you all just have beers together? This was pre-COVID, so you could meet in person, or did you talk on the phone on a regular basis? Or did you fight? I mean, how did it work?
Yeah, it worked wonderfully. And I feel like it, from a standpoint of foundations, I think it was a very innovative way to think about getting people involved. I know that in some other cases, foundations collect a lot of money for research and then basically put out a funding call and say, if anyone wants to work on this disorder, um, we have some money to fund people. But oftentimes, if people are not actually thinking about that disorder, they still might not, jump onto that. And so, I think for some foundations, it's still hard to actually find the researchers. And so, I think this was very innovative that they actually collected this group and put it together. Um, Now, I was used to NIH funding, and NIH generally asks us for a summary of what we did each year. So we have to put in an annual report on what we do. Uh, FAST did things very differently. We had weekly calls where we were talking about it as a science team, but also members of FAST would sit in on those calls. And so we would all be talking about what was going on very dynamically. And I would say it was very fulfilling to be part of that team. Uh,
So did you have, when you said you had weekly calls, so was this where you would actually have evidence every week to talk about on a call, or was it brainstorming? And how were the patients involved, or the families of the patients involved? How did they contribute?
so it was mostly, yeah, presenting the data or brainstorming or whatever, issues we had to tackle those days. Frequently it was, when are we going to see those results kind of thing. And that's also good, because that kept up the pressure to not just talk about things, but actually get things done. And so I think that was important. I'd say the family's interests were represent, or the family voices were mostly represented from people at FAST. But of course, FAST had the gala every year, and so we started out actually giving science talks at the gala to the parents. And I think they quickly realized that the kind of nuanced scientific details that are a big concern to the scientists were often just very hard to communicate with the parents, and that maybe the scientists were not always the best speakers to represent that work. So I think we all learned a lot about science communication and how to do that a lot better in that process. But let's say, I don't know, I hope that the families found value in our presentations to them, but the scientists very much benefited from interacting with the parents. And I would say that that has been probably one of the most impactful kinds of events in my entire scientific career. Um,
Wow, why?
you know, um, I think up until that point, I didn't really have um, a strong focus on neurologic disease. We were trying to apply our kind of gene editing tools in different ways. But it occurred to me at some point that someone could be studying cancer in my lab and never have to meet a patient living with cancer or a family living with cancer. And the personal impact just for me as I started to get more involved with families and get more involved in the community, it was overwhelming. And you know, to talk to the people who we're serving in this role as trying to develop therapies has really had a profound impact on me. And so I've tried to involve the rest of my lab in these kinds of experiences. So we've had angel families visit our lab, and the very first angel family to visit our lab was Mady Dunnigan, and she brought Jackson around. And she she can be very convincing on the phone, and she told us she was going to bring Jackson over, and we met Jackson a number of years ago now, and again that had a very profound impact not just on me but the other people in the lab. Um, and since then we've had visits from other families as well, and we certainly welcome that. We're very happy to receive angels, and we give them a tour and everything, and that's great. But uh, again, aside from just being able to give something back to the families, I think uh, meeting the people that you're working for, it just has a, a huge impact on on just everyone in my lab. Just the feeling of what they're doing and why they're doing it. It really changes things a lot.
I guess it takes it from just the dish and the test tube to an actual child and a family, and that really makes it matter. I It's so hard sometimes. I mean, I know for personally, when I stay up late and I'm working on dishes and dishes of cells and I'm repeating things and something doesn't work out and I have to do it again. And oh my gosh, it can be so discouraging. But if you really think about the person who the cells came from or who the treatment might help, then it keeps you going till 2 in the morning and you get the experiment right.
That's right. That's right. And if I could be candid with you too about that, I feel like I've been involved with this community for a while, and I would say, what I want to do is I want to be able to make therapeutics that help Angelman kids and their families, and it does weigh on me that I haven't done that yet. And it's something that I'm very aware of, and it weighs on me every day. I would say to have these interactions with the families, and I feel like every time that I get that I apply for a grant to work on Angelman syndrome, I feel like I'm kind of making a promise to the Angelman community about what I'm going to do, and I haven't gotten there yet, and uh, that definitely it keeps me motivated. It makes me humble, but I do want to say that I'm not going away, and I'm not giving up. And my intention in all of this is I won't be happy until I've actually helped somebody.
This is why you're beloved, the beloved Dr. Siegel. So, well, okay. So tell me then, what are you working on now? Because it's different than what you were working on before. And is that because what you're working on before you felt like it wasn't going to help? It wasn't going to help as much as you wanted it to, and that's why you're doing something different?
That's kind of right. So the first kind of technology that we were working on was a technology that could turn off gene expression. And the thing that we wanted to turn off was this so-called antisense transcript. That is the thing that in the body keeps the paternal copy of UBE3A from being expressed. And so we knew from a genetic point of view that if we could come up with a protein that could shut off that so-called antisense transcript, that UBE3A would come back on. And we were able to show that that worked, and we published that. However, over time, it became apparent that that might not be the most ideal approach because there's other kinds of RNA molecules that are made in the process of that antisense transcript. And so shutting off all of that, uh, it might have other effects on the brain that we weren't expecting. Now, we don't know if that's completely the case, and we don't know how severe that would be, if it would be a problem at all. But in principle, what we know about that region, it would be better, let's say, to have a therapy that didn't shut off everything there, that really just shut off the part that was interfering with UBE3A. And so consequently, I'd say across the field, um, uh, there's been a real push to come up with kind of molecular therapies that only affects the part of the antisense transcript that affects UBE3A. And so the hope is that by using these kinds of approaches, um, we can let whatever normal biology normally happens in that region, but we just um, affect it just enough so that UBE3A can be turned back on, and we don't affect other molecules that need to be there. So our lab is working on that in one way, which is to use a member of the CRISPR-Cas family called Cas13. And Cas13 is a special member. It doesn't actually cut DNA, but it cuts RNA. And we're using this special property to attack that antisense transcript and chop off just the piece at the end that interferes with UB3A. And we've been able to show, at least in mice, that that approach can actually turn on UB3A in the brain.
Wow. So, okay, I want to rephrase this a little bit. So, the way I understand it, um, there is the way UBE3A on the paternal gene is working, which is what we care about with Angelman, because the one on the maternal side is missing or mutated, and so it's not there. So, we're trying to work on the paternal side. So, the way I understand it is that uh, the gene is going along transcribing, and there's an antisense transcription happening at the same time, and they somehow crash. And when that crashes, that's what makes the UBE3A gene not finish transcribing. And that's why the paternal UBE3A gene doesn't ever get made, right? Okay.
Right.
So in the way so the way you just said it sounds like that there's just this one piece hanging down from that antisense, and that's the part that does the crashing. So if you cut that off, then it can keep going. Is that how it works?
Yeah, I really like your hand diagram of what's going on there. And I guess the best analogy is we're not going to chop off your arm. We're just going to chop off your fingers.
Nice. So I didn't realize that it was just one little piece of the antisense that's doing that. I always just thought of it as this giant, train engine, which is the picture we've seen in presentations, like hitting head on with the other train engine that's going locomotive that's coming through. But it sounds like it's more like a tripwire.
Yeah. So there's a lot of interesting molecular biology going on at that location. And it turns out that by just clipping off the last bit that is interfering with UB3A seems to be enough to get UB3A to come on without affecting the other parts.
That's
And so that is, sorry to interrupt, but that is the approach that the ASO therapies that are now in clinical trial are all taking. Probably some of your listeners will have heard about things like shRNA or microRNA. Those approaches are largely aimed at that same kind of intervention. Our Cas13 approach is also aimed at that kind of intervention. And even some of the more exploratory things that are coming out of Mark Zilka's lab and Jim Wilson's lab are also aimed at the same kind of idea.
So what if all those things are doing the same thing? What's good about the CRISPR-Cas13? Why would I want that instead of an ASO or some other thing, shRNA, or would I want it all or both or a combo?
Yeah, that's a great question. So certainly in this stage of drug development, even with ASOs now in clinical trial, I think we have a lot to learn about how these molecular therapies are going to work in people. And at this point, I don't know if we know enough of any of these to say which ones are going to be the sure winners. Are some of them going to work well in some people, maybe less well in others? And then, I've really come to embrace the philosophy of FAST that uh, we probably shouldn't be in the business of trying to back the best horse. We want to back all the horses and then see who wins. And so I think, there are theoretical advantages and disadvantages to the different kinds of technologies. But, uh, the main thing is what's going to happen on the ground, and we won't know that until we really try. And so I think at this stage of the development process, all bets are open. So I could certainly go on and talk about ASOs and shRNAs and these kinds of Cas13, what the theoretical benefits and limitations might be. but I think, we're learning more about how they're actually going to work. And, I think we all look forward to a time when, if these are proven to be safe, that we could actually see them in the context of what they do in people.
Your background sounds like my background. That's what's usually happening at my house. That's okay. I don't know why it's so quiet here right now. Knock on wood. It's probably going to change now that I said that. So, okay, so that is really interesting. And so I have a couple other questions, and then I want to ask you something about the infrastructure work. But just a little bit more about CAS 13. So where is it right now? Is it in dishes of cells? Is it in a mouse? Is it in a different kind of animal? What has it been tried on at this point?
So up to now, we've just been testing it in the Angelman syndrome mouse model. And I want to say that the work that we do in Angelman syndrome now, it's really a collaborative effort between my lab, the lab of Kyle Fink, and the lab of Jill Silverman here at UC Davis. And we'll probably talk more about that when we talk about the infrastructure project. But we're very fortunate to have not just like a lab effort, but a collaborative effort here at UC Davis to work on these things. And so, that allows us to apply our expertise in making kind of molecular therapies, and then working with people like Jill Silverman, who are, kind of world-renowned experts in behavioral analysis in mice, and have their team evaluate, whether this is a useful therapy. And so we also have a rat model of Angelman syndrome that FAST had funded, and we have that at UC Davis. Because the DNA sequences that we're using are a little bit different between the mouse and the rat, um, this approach right now is only targeted to the mice. Um, and and so we're at the stage where we're looking at the most efficient way to inject it so that we turn off um, or attack that antisense transcript in as many cells as possible, and allow UB3A to be turned on. And we're trying to learn about what level it's turned on, to the extent that it's turned on, and more importantly, how that affects the mouse behaviors. And so that also gets back to what is the timing of the intervention? Does it have to be at an early stage? What happens if we do it at a later stage? And these kinds of things. We do right now deliver it using an adeno-associated virus, which is probably the most common type of method for delivering molecular therapy to the brain right now. And that's also very exciting. But we certainly have a lot of questions about like what is exactly the best way to do that. And so we're learning. That's kind of where the stage is now.
Cool. So, you talked a little bit about infrastructure and how it has to do with two other people there at Davis. This is a fast funded effort. What is it?
So the infrastructure project, I would say, is another kind of innovative approach that FAST is taking to um, for the mission of trying to improve the lives of kids with Angelman syndrome. And there are many drug companies, for example, that either feel like they have a drug that might be useful in Angelman syndrome, or they might have even done some preliminary study that showed, or somebody did a preliminary study that said that a drug might be useful for Angelman syndrome. And the question is how do you bridge that gap between somebody saying, I think this drug might be useful for Angelman syndrome, and then actually, producing the kind of data that would be necessary to take that towards a clinical trial, or is it even worth going towards a clinical trial? Um, and so in the past, I think each individual drug company would approach maybe some investigator in the field that they've read about and say, well, do you have the bandwidth to take on another project, with this drug thing? And, the problem with that, let's say one of the limitations of that approach is that all these laboratories are busy. They have projects that they're trying to do. And so, this is another project that comes along. Do they have enough people? If they don't have the people, do they have to hire new people to try to work on this project? And if it's a 1 year project where they're going to work with a company for 1 year, then are they going to let those people go at the end of the year? Do they have to hire new people for the next project? If they get 3 projects, do they need 3 people? And then how are they going to support those people later? So the infrastructure, again, is another innovative approach where basically FAST said, we're going to fund a group of people to work on analyzing, the drugs that come along from these companies. And I would say that not only, is this team assembled with appropriate expertise to address this problem, but the key element in this is that um, we're allowed to hire people for a number of years, a couple of years, um, as long as this project, shows that it's actually being cost effective to FAST and to the community. But the idea is that we would be funded for, a couple of years. And so we could hire people and say, look, if you come to work on this project with us, we can say that you'll be employed for at least a couple of years and maybe even longer if things are working out. But you're going to be dedicated to trying to work with all these companies on drugs that might be useful for Angelman syndrome. And so I think this has really allowed the drug developers to work with us and very quickly establish a relationship and the funding and all that kind of thing. But we have the dedicated personnel to be able to give a kind of independent evaluation of what the utility of that drug could be in the treatment of Angelman syndrome, and allow decisions to be made on that basis as to whether this can move forward towards a kind of IND, preparing for a clinical trial, or in our kind of objective judgment, it doesn't seem like the evidence supports that.
Wow, that's amazing. So basically, it's like creating a team that's ready at any moment for the next discovery to come down the pike and to test it and see if it's something that could really work. So it's a way to, if I understand it right, it's a way to make life easier for any of the pharmaceutical firms or even other investigators or investors who might be willing to work on developing a treatment for Angelman syndrome. It's a way to make that a lot easier. It's a way to make it less scary and expensive. Is that?
Yeah, that's right. That's right. And so we've been approached by companies that have an individual drug that they think might be useful for Angelman syndrome. We've had other groups that work with libraries of drugs, and they want to know if we could, do a focus screen on that library to see if it's going to be useful for Angelman syndrome. And so, again, our job is to have the expertise on hand. So when those things come in, we could quickly get it so that it can get into mice, or it can be evaluated in a relatively short amount of time in a cost effective way, as you say. And so we can get the maximum benefit from those interactions.
That's so cool. The other thing I'm thinking about is how if you have something tested, especially with the Angelman models and the Angelman mice, if you have something tested in different labs, sometimes they would get different results. And part of that's because the models might be finicky or somebody might do something in a different way. And so if a lot of people are coming to you and y'all are a team of experts who's doing something consistently in a consistent way every time, then you're really going to be able to compare apples to apples instead of comparing apples to oranges, and you'd really be able to tell if something's working or not.
That's right. That is the intention. And I should say that, there's also contract research organizations, and they have a certain role to play in this whole drug development portion. But it would probably be a very expensive and difficult way to go if all the drugs that were potentially useful for Angelman syndrome would have to go through a CRO first, as well as most CROs would not have the kind of nuanced knowledge about Angelman syndrome that our team has been able to acquire over the many years of being involved. So I think it's a really win-win situation for everyone involved.
That's so great. So, well, I have taken up so much of your time, and I could ask you a bazillion more questions, but I'll have to save it for next time, and maybe we can have an update on the CRISPR-Cas13 or find out more about what the infrastructure program's been doing. And so anyway, just one last little thing. Are you still running marathons?
um, I am still planning to run a marathon, when now that COVID restrictions are being lifted and I might be able to travel more, I'd like to be more involved in that. I'm just, I'm a, I'm very happy if I can put my recreation to help with Angelman syndrome. So I think Allison Barrett had also talked me into doing the $1 million bike ride. And so I actually flew my bicycle out with me to Pennsylvania to do the 72-mile bike ride, $1 million bike ride for UPenn, to help raise money there too. So it's been really fun. That part of what I do has been really fun. But yeah, and I hope to do more of that. I just really want to close by saying that, I feel like I'm a spokesperson for my group. So I just want to say again, the work that I do is only possible by working with Jill Silverman and Kyle Fink here at UC Davis. And, the whole interaction has just really been very fulfilling and productive. And so I hope that we can continue to serve the Angelman community until we don't have to serve them anymore.
Wow. Well, thank you so much. I really appreciate that. And don't worry, I'm going to be interviewing Doctors Fink and Silverman too. So,
Excellent.
okay. Thank you so much. You have a wonderful day, and I hope we can talk soon.